All right, chapter 3 covers one of the most important topics in biology. That's cells. What are cells? Kind of how do they work, and what are, what kind of things are important about them. I'm going to describe the size of cells, how we know about cells, what's called cell theory, and I'll talk about prokaryotic cells and eukaryotic cells. And so there's a picture of an early microscope there. I used to really like the Far Side. It's really very good biology about jokes. And there down below are actual cells from a light microscope. On the left there, kind of the reddish stuff, those are cells from the human body. In the middle there, those are plant cells. And then on the right, those are bacteria. Um, so this kind of brings me to the question, how large is the average living organism? 50 kilograms, 10 kilograms, or all the way down to zero point 0.1 grams? Well, biologists didn't really know for a while, but now they're pretty sure that the average organism is much less than a gram because it's probably a bacteria. And because they occur in huge numbers in habitats that you don't see other organisms, in soil, in water, even growing in the air. We really didn't know about these until we developed the light microscope. Okay, and there's a picture of microscopes that we might use in biology. This was really a crucial thing. It helped us recognize how organisms work and the extreme wealth of organisms that are small. Remember, the movie showed us Robert Hooke and Anton von Leeuwenhoek. Leeuwenhoek there is on the left, Robert Hooke is on the right. These were two guys that used magnifying glasses and lined them up in little devices to magnify things. And they were both very good artists, and they drew and they contacted others, and they documented things like cells in plants, bacteria, parts of the human body like sperm cells and muscle fibers. And these guys were kind of doing this as a hobby on their own. Van Leeuwenhoek was probably maybe the better guy at it because he was using microscopes to identify good buys when he was buying cloth. He was a clothing manufacturer and merchant. And so to really know the quality of the cloth, he wanted to see it up close to look at the threads. And so he developed a microscope with where he could precisely position things on the tip of a screw, and then it would be in focus. And he drew lots of bacteria, he communicated with other people, and he shared what he knew, and that was the beginning of the understanding of living organisms. And after von Leeuwenhoek, we realized that most organisms are quite small, and even large organisms are made up of small living parts, which we now call cells. Okay, the microscope can see down to below 1 micron. So that's a micron is 1/1000th of a millimeter, so much smaller than the eye can see. And doing this, they realized, oh, there's an abundance of organisms in the environment, and that most of the parts of the human body are quite small. And so, we know what a meter is. That's about 3 feet. That equals 1,000 millimeters. 1 millimeter, which is a very small unit, equals 1,000 micrometers, which is about the size of a bacteria like E. coli. And so that you need a microscope to see that. And 1 micrometer equals 1,000 nanometers. And most viruses are in the realm of nanometers, and so we couldn't even see those. Light microscopes use magnifying glasses, and they can resolve things down to a little less than a millimeter, but it gets fuzzy. And this is because of the size of the wavelength of light. Electron microscopes, shown below here, can magnify much greater because electrons travel in straighter lines. And so we can get to 0.2 nanometers, so much, much smaller, 1,000 times smaller. So we can get to things like viruses and even large molecules. With electron microscopes, often what you have is cells that you'll shine light through them and they'll show up as glowing or twinkling or dark objects against a background that's white. We can stain them to make them look more visible, but that's what we see with them. The electron microscope shoots a beam of electrons at something, and then the electrons are blocked or bounced off. We usually have to coat whatever it is we're looking at in metal to see it. But electron microscopes have very, very high resolution. They can see things that we just can't see with a light microscope. The surfaces of things we see with scanning electron microscopes, that can magnify about 10,000 times. And transmission microscopes, which we can beam electrons through things, that we can see to about 100,000 magnification. So after light microscopes for around for about 100 years, it was recognized that everything we looked at that was alive was made up of one or more cells, and that living organisms really meant cells. And anything smaller than a cell wasn't really alive, and cells only came from pre-existing cells. By this time, they recognized that there were two cell types: a large complex cell called a eukaryotic cell, which had a nucleus, and a small cell, which we call prokaryotic, which didn't have a nucleus, but had DNA in the center. We now commonly call these bacteria, and they're a cell that's maybe 10 times smaller than the eukaryotic cell, but very, very abundant. In all cases, the cell size is very limited. Okay, cells all kind of work the same. They absorb things by diffusion and osmosis, and so that means that to get rid of their wastes, they can't be too big. Otherwise, the waste would have a hard time diffusing out. The surface area of a cell only increases with the square of the radius, but the volume increases with the cube of the radius. And so small cells are just more effective at getting rid of wastes. And so, the diffusion is limited by surface area, temperature, and concentration gradient, and the distance things have to happen. So the first one and the last one are really make it much more advantageous for cells to be small. Okay, so here's a diagram of those two basic cell types: prokaryotic cells and eukaryotic cells. Prokaryotic cells are small, eukaryotes are large and complex. In prokaryotic cells, there's no internal organelles or compartments, no internal membranes. All eukaryotic cells have a nucleus, and often they have many other organelles that are separated by membranes. We have fossils of bacteria that go back over 2 billion years, but eukaryotic fossils are, and they fossilize better than prokaryotes. They only are about 1 billion years old, we think. But all multicellular organisms are eukaryotes. So everything we see is eukaryotic. Prokaryotes, we can't see them, but they are metabolically diverse. They can do unusual chemical reactions. Many of them are anaerobic and doing weird, weird things. The prokaryotes include two groups: the archaea, that includes methanogens that live in hot springs and deep hot water. A lot of the organisms like hot water or salty water. Bacteria contain both aerobic and anaerobic organisms, and both of these are small. In prokaryotic cells, it's very simple. The cell has a cell wall, then a phospholipid membrane just inside the cell wall. There's one big chromosome, circular chromosome, in the cytoplasm, and then the only organelle they have besides that DNA molecule is a ribosome. So very simple. You don't see many structures. They can have a flagella. They can have a capsule, which is kind of a slime coat around them. But there's not too much going on there. There's two organisms, two groups that have this kind of cell: the archaea and the bacteria. I'll give you a couple examples of archaea and bacteria. Actually, this is just one example. These are two different views of Salmonella. On the left is a light microscope view of Salmonella in red, invading human cells. Okay, these bacteria are small, and so they can go inside the cell and reproduce in there. On the right there is an electron microscope picture, and that shows uh, again bacteria invading cells, but here the magnification is much greater. You can actually see the surface of the cell. So there's the nice bacteria on the right. In both cases, there's a size bar. So you see this one has much greater magnification. That size bar is 1 micrometer. Here, the size bar is 25 micrometers. Okay, just for giggles, how many prokaryotic cells are found in the human body? I said we're made up of eukaryotic cells, but actually we have large numbers of prokaryotic cells, trillions of them. Are found on our skin surface and in our large intestine. They should never be in our bloodstream and our nervous system, anywhere like that. But there are parts of the body that have lots of them, and so there's some, not all of them are bad. Not all of them cause disease. Some of them are doing useful jobs for us. They're breaking down things we can't break down, preventing worse bacteria from occupying these things. That's kind of what they're doing. Okay, so next time we're going to talk about eukaryotic cells. These are the more complex cells. On the left there is a plant cell. On the right is an animal cell. All these cells possess a nucleus and lots of internal compartments. Biologists have documented many, many different kinds of cells in the human body, many different kinds of cells in plants and animals. There's lots of application for looking at cells. Microscopes, we have lots of different uses for them in hospitals and research facilities. Here's one application. On the left there are the normal cells of a uterine cervix, a portion of the uterus, and those cells are shed very easily. They kind of come off like skin cells, and those are normal cells on the left, but on the right there are cells that are enlarged and infected with papillomavirus. These are what we're looking for when we have a woman has a pap smear. Some of these are precancerous growths caused by a viral infection of a papillomavirus. So a good example of when we look at a microscope to look for abnormal cells. So eukaryotic cells we'll talk about next time. These include organisms like protists, algae, fungi like yeasts and molds and mushrooms, all plants, all animals, including us. Okay, so we'll pick this up next week and we'll have lots to say about cells.